Thank you very much, and thank you all for coming. I hope at the end of the presentation, I can also thank you for staying. The title of the presentation is just like in a honeybee colony. It takes a team in the UC Davis hive to win an award. And my friends from entomology knows that I use here the honeybees metaphorically because I don't work with the honeybees. Honeybees are very important though, and we know if they disappear from the face of the planet, humans would have only 4 years left. And attributed this quote to Einstein, but the University of Manchester denied vehemently that he said that. But I think everyone knows that Einstein loved the bees. I attended many of these presentations, include today's brilliant presentation by Pam Ronald, and they all have a different style. So I'm going to use a different style. Typically, at the end of the presentation, the speaker thanks and acknowledges people who contribute to their research, but I'm going to give you an extended thank you. So all the slides are going to be thank you to the various people that contribute to that. And if I'm going to acknowledge the contribution of people, I have to start with the family. This is the family in Japan, Beatriz, Auguste, and Gabriel, who were born in Japan. At the time that we moved to the United States, Beatriz was pregnant, and we arrived here, Elena was born. So technically, Elena is like a Subaru that was made in Japan and assembled in the United States. After all, love is what makes a Subaru, right? Anyhow, so I want to thank the family first for the time, in particular Beatriz, for being able to guide the kids through college. We celebrate Augusto in Princeton City in 2013, Gabriel in Hanover 2019, and Elena in Houston 2022. That's how Brazilians invested $600K in the stock market just before it crashes. So Gabriel and Elena contribute to this research, so I want to acknowledge their contribution. Augusto was interested in me teaching some skills that I do not have. So I was not able to do that, so he became an athlete by himself. But Gabriel and Elena did participate in research, and Gabriel was very curious, and he came to the lab when he was still in elementary school.
Hi, I'm Gabriel Leal, and I'm a student in Leslie's Whitebird class in Leal Elementary.
So he did a project there for his class, and there was no publication related to that. But later on, when he was in high school, he came to the lab and we published this paper together that it became cited as many as 38 times. And even recently, he came to the lab to perform one task in our research that we are not able to do, and he has the skills to do that. Elena came in very early. She could not even hold the syringe to inject in the chromatograph. But later on, when she was in high school, she came to the lab with a very important question that many of you might have. Why I am getting more attracted to mosquitoes than other people? And she wanted to address this question, and we worked for that in a research project that she ended up publishing that research project. And they also give a presentation at the University of Brasília, and also here at UC Davis.
So today's speaker is Leal, and she's actually from Davis High School.
I'm still a senior in high school, so go easy on me.
So the question was why she's more attracted to mosquitoes than other people. So at that time, we had two undergraduate students that also participated in this project, Justin Huang and Kai-Ming Tan.
Hello, I'm Kai-Ming Tan. I was in the Leal lab at UC Davis from 2015 to 2019. I'm now a family medicine resident at University of Pittsburgh Medical Center.
It's very nice to see these students grow. And what Kai-Ming and Justin did, it was they record the response of mosquitoes. And Lennon was on the other room where the smell of the hand, one hand was sent to one side of the olfactometer, and the smell of another hand that was covered was to the other side of the olfactometer, so the mosquitoes decide which side to go. The mosquito indeed went to the side that comes from her smell, so they like it. Then the next thing we did was to compare an attractive to a non-attractive person, and surely enough, mosquitoes came preferentially to the side that's depicted here in blue, represented that she is more attractive than me, for example. Then the next question was, what makes this attraction? What's the chemistry associated with that? For that, we have to collect the compounds from the skin of the person. And she's my daughter, so the IRB was very complicated. Thank you, Dr. Maverax, to help with that. And finally, we got the permission to do this research, and we figure out what's the chemistry. There are 3 aldehydes, basically, and the proportion of these aldehydes, the ratio, is what makes the big difference. But at that time, we found another answer for another question that Elena had for many times. She always said, the bees are always bothering me. And when we collect the volatiles from her, we note that there was a contaminant, isoamyl acetate. Isoamyl acetate, to make a long story short, is a chemical that the bees elicit a very aggressive behaving bees. It's called the sting pheromone. It's known from the early 1960s. And we figured out that that chemical was coming from a shampoo that she was wearing at that time, the so-called OC. So she stopped using that shampoo, and there was no problem with bees anymore. In terms of acknowledgement, we have to acknowledge the funds. The funds, like Jeremiah Wild used to say, without money there is no music, without funding there is no research. I would like to thank the National Institutes of Health, the National Institute of Allergy and Infectious Disease for the continued support to my research activities, and a more recent fund from the CDC under the leadership of Chris Barker in the PAC-VECT program here. And my own funds from the National Science Foundation, USDA, Funda Citrus, Biochemistry Bio, Almonds and Pistachio Boards, and the various donors. There is one donor in particular that I want to acknowledge. Dick Rice. He gave $30,000, $30K, towards my startup here at UC Davis as an anonymous donor. And one day I needed to buy something. I went to the office, and Carmen said, I need to call Dr. Rice. And then I figured out, oh, that was Dick. Then I met Dick at the Entomological Society of America meeting, and I asked him, you are the one who gave this $30,000, right? And he just smiled. Thank you very much, Dick. We miss you a great deal. I would like to thank the administration. I'm very glad that I put that slide in here because the provost is here. We want to acknowledge that during this period of time that I'm here, the research enterprise at UC Davis increased. It was about $250 million, $250 million a year of research funds. Now we are in the excess of $1 billion. So we are increasing as a research enterprise. Back then, this type of award had a big ceremony and a sit-down dinner, and the chancellor would come and give a medal. I'm waiting for the provost to give the medal after my presentation, if you can afford to stay until then. I would like to thank the staff. The staff is essential for our work. We always thank the people who did the research or something, but we forgot about the staff. And I wanted to mention 2 members of the staff that epitomize how much the support they give to us. I spent half of the time here in entomology and half of the time in MCB so far. So I want to acknowledge Kathy Gavi that epitomizes the support that we get from the staff. She's always behind the lens. We can't find Kathy Gavi in front of the lens. She's always behind the lens, ready to support any research, advertise any research that is related to entomology, especially if it's related to honeybees. That's why I put the title honeybees to call her attention. And the staff from MCB. Everyone there is fantastic, but I wanted to show an example as Heman Patel, the TI guy. Sometimes we have problems with computer midnight, or sometimes during the weekend we send an email, expect that we're going to get a response in the next Monday, like 9 o'clock. In 10-15 minutes, we get a response from Heman. Looks like he works 24/7, but it's not paid that much. Always there. Most of the time, his major recommendation to solve most of the problem is the following.
Reboot your computer.
Anyhow, so he says, reboot your computer 9% of the time, he solves the problems. I would like to thank all the students, visiting scholars, postdoctoral scholars, research scientists, and collaborators. My friends, when they see my papers, there are too many co-authors, they call that my papers have a cast of thousands. I try to give credit to everyone who contributes in some way to our research activities. There are many of them that I cannot talk about their work today. I don't even have any photos of all members of the lab. But these are some snapshots of the website throughout the years that highlight some of the people that were here at different times. They all contribute significantly to my research activities and to this award, but I do not have time to talk about the work of all of them. The lab started here in April 1st, 2000. Yes, April 1st. And I didn't name the lab after my family name, which is a convention here. I named it after two colleagues from entomology, Susumu Maeda and Sean Duffy. So the lab was called the Maeda Duffy while I was in entomology. And Sean Duffy passed away and vacated the position. The FTE was a replacement FTE. Back then, the FTE would go to the department would hire again. So technically, I am a replacement for Sean Duffy, but it's better to say FTE replacement for Sean Duffy because no one would be able to replace Sean Duffy. I came to UC Davis, and I had my tenure position in Japan, and the two faculty members here from UC Davis make a very big decision in my mind to apply for this position here. One was Bruce Hammock, who came all the way to Japan, to Tsukuba, and he came to my lab to try to convince me that I should apply for a position here. This is about the time that Bruce got elected to the National Academy of Science. And another one who was visiting there in Tsukuba, and he came to my lab to say how great UC Davis was Paul Getz, who was then the chair of the Department of Agronomy and Range Science. They all said these great things about UC Davis, so I applied for the position. A few months later, I got the letter from the chair of the search committee. Thank you very much for your application. After careful consideration, when you hear this phrase, after careful consideration, don't even bother to read the rest of the letter because it's a rejection, right? And I said, okay, I have my position here. I keep it going. 1 week later, I got a phone call from the department chair and they said, we want to interview you for the position. In my mind, I asked, do they talk to each other? Anyhow, so I came, I organized the trip, and they then came, and they are supposed to meet me at the airport. When I arrived at the airport, there was no one from UC Davis to be seen. So I called Beatrice to tell her that I arrived safely. And I told her, maybe the chair of the search committee is the one who is right. Anyhow, so I was very relaxed. And then I interviewed for the position. I got the position, and everything is history. The lab started very small with two postdoctoral scholars. One was Yuko Ishida from Japan, who played a very critical role in me receiving this award today. Yuko Ishida, who is now back to Japan, working for the Rachojima Recreational Organization, the Galapagos of Japan.
Hi, I'm Yuko Ishida.
Who remains a very strong fan of the king. And the other one was Good Schuller. Good Schuller was able to visualize the goals of the lab in the website in this animation. The goals of the lab are understand chemical communication, chemical signal, for example, emitted by a female to attract a male insect, and the proteins that the males utilize in the olfactory system to detect those compounds. So those are the proteins that we call odorant binding proteins, odorant degrading enzymes, and odorant receptors. When those proteins are related to a pheromone, we call them pheromone binding proteins, pheromone degrading enzymes, and the pheromone receptors. So these compounds, these pheromones, are compounds that do not dissolve in water. And inside of the antenna on the sensilla, before reaching the receptor, there is water around the receptor. So these molecules need a boat. So they ferry the molecules to the receptor, and when they activate the receptor, there is another protein that goes there and degrades the chemical signal so that they cannot keep activating the receptor over and over, depicted here in this green cartoon. Early on, we discovered that the pheromone binding protein from the silkworm moth has two different conformations. One is that it binds to the pheromone, and one, a low pH, that does not bind to the pheromone. We thought at that point that this might be the mechanism of pheromone bind and release. They bind, carry to the receptor. The surface of the receptor dendrite is negatively charged, so there is a accumulate of protons that is a localized low pH. So we thought, well, that might be the way that they are delivering the pheromone. For that, we collaborate with structural biologists to understand better the structure of the proteins, and we collaborate with John Clark from Cornell, then moved to Harvard, and Kurt Wutterich for ETH, who is now in Scripps in San Diego. John Clark got the crystal structure of the protein bound to the pheromone, I see here on the left, and Kurt Wutterich found something very, very interesting for us, that the C-terminus of the protein get as a helix and penetrate the binding pocket, thus provide a blocking for the binding pocket and release the pheromone, ejecting the pheromone. So we knew there is a conformational change, but the question that we had was, is this conformational change fast enough to justify what happens in real life? The insects are flying, and they have 50 to 100 milliseconds to make a decision, what is the next point that I go to fly? So they have to detect the smell and get rid of the signal within 50 to 100 milliseconds. So the question is that, is this conformational change fast enough for that? If it's not, does not justify the mechanism. By then, the lab was already growing, and I thank very much one student, Vic Xiang. Vic Xiang is one of those undergraduate students that display a nucleation effect. She came in after her, many undergraduate students come to the lab, including Angela Chen.
Hi, I'm Dr. Angela Chen. I'm an associate professor and a clinician at Marshall P. Ketchum University. I'm a UC Davis alum, and I work in Leal lab from 2002 to 2004, including 1 year as a postgraduate researcher.
She was a very productive collaborator in the lab, and with Angela, we addressed many questions, including we measure for the first time, use stop-the-flow fluorescence, the half-time of this reaction. So the uptake of the pheromone is in the order of 1 millisecond, and then we also found out that the pheromone would be delivered very slowly, if not for the conformational change, that speed up the process to 90 milliseconds, the half-time. So basically, the conformational change what make the pheromone to be delivered and to be processed in real time. So that was the first time that we measured the kinetics, and we published this paper in the Procedures of the National Academy of Science, and it has been cited 223 times. I mentioned Kurt Wutterich. He's the man who invented the technique for using NMR to determine the three-dimensional structure of protein. He was already eligible to get the Nobel Prize, but we know that to get the Nobel Prize is not sufficient to be the best in the field. He needed something else. When he put in his CV collaboration with Walter Leal, then he got the Nobel Prize in 2002. We celebrated that, and 10 years later, we both got elected to the Brazilian Academy of Science, and here's the picture for the induction in 2013. At that time, I made T-shirts for the group that reads, I did the work, and Walter Leal got in the academy. Anyhow, that induction was a very long one, because the late Minister of Science and Technology, Raup, he gave a very long speech. So it gave time to me and Kurt Wutterich to write basically the next manuscript that we published in the Procedures of the National Academy of Science, showing that we had prepared the protein for him, and the protein was truncated, and without the C-terminus, the protein that binds the pheromone and never releases anymore. And also we show that it's very important, this point, some of the ligands might never be dropped from the protein, and some ligands might be dropped earlier on so that the pheromone degrading enzyme capture that. We did a lot of work in agricultural pests. Navel orange worm is a very important pest of almond, pistachio, walnuts, everything that makes money in agriculture. This is a pest of. So navel orange worm, we start on that because we talked to Frank Zeylon. If you want to do anything in entomology that's important for an agriculture, you have to go talk to Frank Zeylon. And Frank said, well, there is a problem with navel orange worm. The pheromone was known, but it didn't work, because it was missing something. Pheromone normally is a combination of various compounds, and at that time, everyone knew that the pheromones of Lepidopterus are aldehydes, acetate, and alcohol. And everyone was focused on that for 30 years. Then we came to the scene and we solved the problem thanks to one student in the lab, an undergraduate student, Ana Lia Parra Pedrazoli from the University of São Paulo, campus of Piracicaba. She analyzed the response of the male antenna to the pheromone of the female, and she found two peaks that always very small peak, but very consistent. When she told me that, I went after the compounds and I broke down the structure by doing chemical derivatization and ended up that they were not aldehydes, acetates, and alcohol, but highly unsaturated hydrocarbons, which was not known as pheromone at that time. And now, after this publication, many other people found the same similar things. The issue here is not the compounds are complex. Compounds are very simple, but they are very small amounts and they buried in a big mixture. So it's like finding the needles in a haystack. One technique that helped me at the end to identify and to nail down the stereochemistry was to say, to use the GCIR that shows that basically all the double bonds are in the cis configuration. We published this paper and it has been cited 104 times, but these citations are not the most important thing here. We save millions of dollars to California agriculture because now with this pheromone that's work, now they are utilizing this pheromone in the field with the natural compound to reduce the population, so they put a high concentration of the pheromone in the field. The male cannot find the female, and they do not mate, so therefore they reduce the population of the insect. With that, they reduce also the use of insecticide, which is expensive, and also it's a good thing for the environment if you reduce the amount of use of insecticide. Here's the trap. The pheromone was synthesized by Biochemistry Research Incorporation. Here's Frank Zaylon checking the trap, and here's Helena when she was small, also checking the trap and having fun, and wear a t-shirt of Penn State. And thank you very much for Bobby Washino for not letting her wear this t-shirt for a long time. We had some other works in the Naval Joam, and one was done by a student, a graduate student doing the master program, Kevin Clunan.
Hello, I'm Kevin Clunan.
And he did some research. Here's what he did, and he was explained to his peers in 2012.
And then I'll focus on the research, which is the oviposition deterrent for the naval orangeworm as a pest of pollens.
So he completed his master's degree here, went to Penn State, got his PhD there with Tom Baker, and now work at the USDA as a research scientist. Another student that had displayed a nucleation effect, Wei Zhu.
Hi, I'm Wei Xu.
Wei Zhu went back he's in Australia now, and after he came in here, I got so many applications from students from China, very good students after Wei Zhu. Here he collaborated with Jimmy Ames and the others, and they did many projects. One of them, they found that the navel orangeworm pheromone binding protein has this helix that goes in the binding pocket, and they found that there are two histidines there that make salt bridge. And when the pH changes, we know that histidine changes the charge, and for that it can release the C-terminus or can keep lock the C-terminus inside of the binding pocket. And we look at the various pheromone degrading enzymes. They are important. We want to focus on two insects, a very important invasive pest, Popilla japonica, the Japanese beetle, and a species that we have a lot of information, from basic information, that's Antareia polyphemus, very people's studies, a model insect. So I wrote a grant to identify the pheromone degrading enzyme for these two species. The grant was rejected. The main justification was because it was over-ambitious. And I said, over-ambitious for you, not for me. So I separate the grant in two, and I rewrote the grants and new goals and everything. And one got funded by USDA, another one got funded by NSF. Thank you very much to the reviewers for that comment. So anyhow, we isolated the first pheromone degrading enzymes, and for that work, Yuko Ishida played a very, very important role. He found out the precise time where we can go and extract the pheromone degrading enzymes at the maximal amount. We did that, and then we isolated the pheromone degrade enzyme using the same techniques that we teach in BIS 102, can use all the process of gel filtration, ion exchange chromatography, and so on. And at the end, there were one impurity that could not get rid of that. So I have to use the IEF, isoelectric focus. And you see here there are two bands. One is an empty arrow, and another one is a solid arrow. The solid arrow is the target, and the empty one is the impurity that we could separate easily in the IEF gel. But IEF gels are very fragile, and I broke the gel in two pieces, and the band was just in the middle there. As you see, the band there is broken. Instead of crying, I just send one band to one vendor and another one to another one to get the internal sequence of that. By then, in-gel digestion was a thing, and I got the sequence from this company from the East Coast, and the sequence was like that. So this was a nightmare because there are so many uncertainties there. There are many amino acid residues that has degenerate codons. To design the primer for PCR was a nightmare because the combination was enormous. And I said, there is only one person that will be able to do this work is Yuko Ishida, because he's Japanese and the Japanese never say no. So I asked Yuko, you like to do this work? He said, yes. Okay, let's do that. And then I expected that maybe 3 or 4 months later, Yuko was going to come with some results. But within 2 weeks, he sent me this email in the middle of the night.
At 10:19 p.m., April 2nd, 2004, Ishida wrote, Dr. Leal, thank you for waiting for 3 years. Finally, I got a cDNA fragment encoding the esterase. Although this is a preliminary analysis, I confirmed a conserved esterase amino acid region and our identified amino acid sequence in the translated amino acid sequence. I strongly believe this is a Paul P. D.E. Ishida.
He said, I got the sequence. And then I was surprised, and they said, Yuko, that's interesting because the sequence that you show me, it has one residue that I see, there's one segment that I see also the sequence that I got last night, the night before for that. Then I told, go ahead, Yuko cloned the entire sequence, expressed the protein in baculovirus, and together we did the same purification that we teach here in BIS 102. And at the end, when I look at that, I said, my goodness, this is perfect. The recombinant protein is identical to the native protein. So I asked Yuko, how in the world did you get this thing in 2 weeks? Because there are so many possibilities. And Yuko told me, I had a dream. And in the middle of the dream, I saw 2 primers. And these were the first pair of primers that I used, and it worked. So I said, okay, it works for me because the sequence is perfect. So sometimes people see Yuko like that in the lab, and they would come to me and say, Yuko is asleep in the lab. No, don't bother him. He's designing primer for PCR. It was a very productive time. So we got analyze all the parameters of the enzymes, and compare the native and the recombinant enzymes. They are identical. So we got finally, for the first time, we got a cover of PNAS for this article. We did the same work for the Japanese beetle. The Japanese beetle had also some excitement because the pheromone has an antagonist as an antipode, and the antipode is a behavioral antagonist. That was known since 1977 by the work of Jim Tomlinson. Then we found that the enzyme degrades this pheromone very rapidly, but the behavioral antagonist more slowly. There was another publication that we published on this work in the Proceedings of the National Academy of Science. For that, we have to invent new ways to measure the degradation, and so on and so forth. No, it was not overambitious. Anyhow, this is UC Davis. Our first chancellor is a medical entomologist. So medical entomologist is in our DNA. There is even a species of mosquito named after our first chancellor, Anopheles freeborni. And that's a species that is vector of malaria. And the West Nile virus came to the United States. So it was time to shift a little bit from agriculture to look at medical entomology. So I started to look at the mosquitoes because they are very important. When they get a bloody meal, they may be contaminated with some virus, for example, West Nile virus. And after a certain time, this virus is going to replicate in the mosquito gut, and after sometimes they bite someone else, they are going to transmit this virus. So they have this very sophisticated apparatus that they pierce your skin, and they go inside and they can find your blood vessels and suck your blood. And they have this very interesting property that they can suck the blood and spit at the same time. And the spit the saliva may have some virus. So they suck the blood and spit, and yes, the mosquito sucks. Then they can transmit various diseases. There's lots of information here. I don't have much time today, so I made a separate video called the supplemental information that I'm going to give Emily to pass to you later on. But there's one work of mosquitoes that I wanted to mention. We look at the odorant binding proteins from the yellow fever mosquitoes. We express these proteins, and we collaborate with Professor Glaucius Oliva from the University of São Paulo to get the structure of the protein. We found very interesting things, pH-sensitive lead and so on. But what I want to say is more important than that. Glaucius Oliva later on became the president of the National Science Foundation equivalent in Brazil called CNPq. And that time, Brazil launched the Science Without Borders. And then we have a connection here, Professor Olivas that became president, and boom, we brought him here thanks to this mosquito oviposition odorant binding protein and to the leadership of Bill Lacey that has a vision to understand that, and UC Davis was the first institution in the entire world to sign a contract under that program. And our contract became the model for many other places, and Bill Lacey, being Bill Lacey, he looked at the Brazilian contract and showed to the Chinese and said, look at what the Brazilians are doing. And the Chinese said, if Brazil is doing that, I'm going to do that as well. So we got lots of results from this, and we brought lots of interesting people here, a diverse group of students. We had many wonderful collaborators and postdocs in the lab. One of them, Zain Said.
Hello, I'm Zain Said.
So Zain Said knew that the birds are the reservoir of the virus. So they knew that he knew that the birds carry the virus during the winter, and then when the population of the birds decrease, there is no birds there, mosquito goes after people. Then Zain asked the question, what's the commonality? How do they find the birds and they find the people? And then to make a long story short, he analyzed the chemistry emanated by the birds and by people and compared the two, and he found it is very simple here, no. Yes, that's true. There's such a job. Next time that you complain, I don't like my job, think twice, because there are such a job. Anyhow, so he found no, very simple compound, and he found the sensor in the antenna of the mosquitoes that was extremely sensitive to this compound, no. We are interested also in repellents, that major thing, and Zain found for the first time that there are sensors in the antenna of the mosquito that respond to DEET as a smell like any other compound. So there was this hypothesis at that time that DEET is suppressing the emission of the chemical signal from the skin. And we make these tests and we demonstrate that's not the case. The case is that they have a sensor, they have a neuron on the antenna that responds specifically to this DEET compound. So we published it, and then this work appears also in the New York Times. We went after many receptors. Receptors each insect has a huge number of receptors, maybe hundreds or so, and one single co-receptor. And they make a pair of a co-receptor with one of these receptors, which becomes the ion channel to detect the chemical signal. We look at those, and we did various collaborations, one of them with Deborah Kimball, who is here today. Thanks, Deborah. And we used the so-called empty neuron. That was a technique developed by John Carlos at Yale. And this flies has everything, but it's missing a receptor. So Deborah, with the genetic manipulation, was able to put not only one type of fly that have the receptor from the moth, from the silkworm moth, and another type of fly that have the receptor and also the pheromone binding protein from the moth. We compared the two, and they do not differ significantly, and then we refute the hypothesis at that time that they do not need the pheromone and the pheromone binding protein to activate the receptor. Only the pheromone itself activates the receptor. There is much more to this story, but I don't have time. And also there is a subsequent collaboration that involves archeon copy, but I talk about that in the supplemental video. We discovered the first receptors for oviposition attractants in collaboration with Chuck Luege from the University of Miami. He's a specialist in 2-electrode voltage clamp and discovered the first receptor for skatol and for indole, which are oviposition attractants. Then I hired Ping-Shi Xu.
Hi, my name is Ping-Shi Xu. Walter recruited me when I was down. During the 12 years working in his lab, he gave me much inspirations and the freedom on the research programs.
This was an amazing 12 years. Ping-Shi just went back to China to take care of his parents. He not only produces so much, but he also educates so many people, and he taught this technique to so many people that went back to their countries now, and they are utilizing this technique in their own laboratory. One of them was Fan Zhu.
Hello, I'm Fan Zhu.
And the Fan Zhu, together with Ping-Shi and the others, they discovered the first pheromone that responded to this compound, 4-ethylphenol, and then they demonstrated 4-ethylphenol is an oviposition attractant. And we don't like things only in the lab, we would like to see things in the field, so we collaborate with Dr. Rosângela Barbosa.
Hi, I am Rosângela Barbosa.
Who did the experiments in the field and confirmed that they indeed are oviposition attractants. And the Fan Zhu at that time knocked down that receptor and they show a direct link between the receptor and the oviposition behavior. Collaboration with Dr. Carlos Vieira, we're very lucky to have him here, and many others who discovered the various receptors. And one of those receptors, Yang Mu-Chu worked on that.
Hi, I'm Yang Mu-Chu.
Yang Mu-Chu discovered the receptor, that most sensitive receptor of all that we have for this insect, for a compound, a very simple compound, acetaldehyde, that we have no idea that this compound has anything to do with mosquitoes. And Yang Mu, together with others, including Justin Wang and Fan Zhen, they demonstrated that this is an oviposition attractant, and we tested this compound in the
Hi, I'm Fan Zhen.
Fan was part of this collaboration, and they also had her own research project that has many publications, including this one, where she shows that some perfume that we wear, some perfumes had compounds that are repellent to mosquitoes. So some perfumes are repellent to people as well, right? But there are some perfumes that are repellent to mosquitoes. When we discovered Ping-Shi discovered the pheromone, the receptor for DIT, we wanted to have the cover. This is not a cover, this was a suggested cover to PNAS, and we gave this presentation that was, ladies and gentlemen, we got it, the DIT receptor. Because everyone was looking for this receptor, and we got it. Ping-Shi showed that the receptor responds not only to DIT, to PMD, picaridine, IR3535, all the commercially available repellents, and he also demonstrated that this responded to this compound in here, methyl jasmonate. So we didn't know why DIT, because DIT is a synthetic compound, but thanks to this research, we then now know that they respond because they mimic a natural compound, methyl jasmonate. So we got the work published in here, and here's the DIT team with Ping-Shi, Yang Mu, and Alisa De La Rosa.
Hello, I'm Alyssa De La Rosa.
Who is now a senior environmental scientist in the Department of Cannabis Control here in the state, and she played a crucial role in this project as well. Most insects respond to DIT. Cockroach, you name it, it's not only mosquitoes. There is one insect that does not respond to DIT. It's the Rhodnius prolixus, the insect that carries the Chagas disease. We had a collaboration with Dr. Ana Mello.
Hello, I'm Ana Mello.
And she sent here her graduate student for a sandwich program, Tiago Franco. And we were looking for the receptors for the pheromone. And then accidentally we discovered a receptor that responds to some compounds, and these compounds are repellent to Rhodnius prolixus. So accidentally looking for a pheromone receptor, we found a receptor for compounds that are repellents, and they are the first repellents. When Zika came became a problem, the pandemic started, we not only created awareness through the symposium, but also I brought my lab to Brazil. I brought the lab there because in collaboration with Rosângela Barbosa, with Constância Arias and the others, They have the mosquito, they have the virus, they could infect the mosquitoes, and we could do the research there and measure the behavior of the mosquitoes. And then we address this question: does the Zika virus in fact affect the mosquito response to repellents? And the answer for this question is no. Thanks to God is no, because if it's yes, then we would not have a repellent. However, we discovered something very important, that when the mosquito gets old, because they need time to replicate the virus, when they get older, they get less sensitive to DEET, and we didn't know that at that time. So then we learned that in order to fend off mosquitoes that are infected, old mosquitoes, we have to use higher concentration of the repellent, like 20-25%. The industry pick up on that, and immediately they put on the market the repellents now that have 25% of DEET. So if you are going to buy some repellents this summer, and you find they're 7%, that's very good for repelling mosquitoes that are not sick, okay, not carrying disease. But if mosquitoes that are carrying disease, we need more. We need 20 to 25% of DEET. Last but not least, Flavia Franco.
Hi, I'm Flavia Franco.
Flavia Franco took here a very important task to find out what's going on with these two receptors. He came to us from the University of São Paulo, campus of Piracicaba, where I have a collaboration, long-time collaboration with Professor Mauricio Bento. These two receptors, one responds to one compound, another responds to the other compound, and they have the inverse specificity. So she looked at those two receptors, and she found that there is only one amino acid residue that make one receptor switch to one to the other. Although they have 300 some amino acids, only one of them is necessary. When we found out that, then we start a collaboration with Vladimir Yarovoy, and Vladimir recruited his students, his graduate students, Brandon Harris, and together we studied that, but I will mention that more in the supplemental video. But I want to show you what Flavia did. These are the two receptors on the left. To make it simple, that looks like that. The receptor on the left is for the red, and a little bit responsive to the blue. And the receptor on the right is a response to the blue, a little bit to the red. And with a single amino acid residue, they switch, they become completely different, opposed, like that. It's like if you talk to a Democrat, and with a single argument, that person becomes a Republican. Or if you talk to a Republican, and one single conversation, you make that person becomes a Democrat. It's not going to happen, but in mosquitoes it does happen with a single amino acid residue. So I would like to thank you all for your patience. I would like to thank the staff of the Academic Senate for putting this thing together. Emily in particular, she's sensational in terms of organizing everything. I would like to thank Bruce Hammock for nominating me for this award. And I hope that I convince you that the work performed by this cast of thousands deserve the Distinguished Research Award. Like Amity mentioned, the National Academy of Science announced last week the new members. Dinesh is there. And then they say here, Walter S. Leal. I'm Walter Leal, and I approve this message. And anyhow, so this was interesting. And then we got, when they get elected to the National Academy of Science, we get so many emails and phone calls and everything. And there was this letter from Kurt Wutterich. And here what it says.
I continue to have fond memories of our highly successful collaboration for more than a decade in the 21st century, as you certainly recall that the structure of your Bombyx mori pheromone binding protein, BMPBPA, was the dominant centerfold of the 2002 Chemistry Nobel Prize poster. I don't think it needs more to document that your proteins were ideal candidates for the use of our NMR techniques.
This is the poster advertised the Nobel Prize, and an entomologist cannot be more proud than that to see the protein that was generated in our lab to be there. I made a joke about him got the Nobel Prize by my CV, but indeed his poster presented this protein, so highlight how important the discovery of this conformational change was. Thank you very much. This is the aphorism that in the door of my office, late Professor Charles Jazz gave it to me when I arrived here and said, you might need this, and perhaps it is working. Thank you very much.